And we're going to start in just a minute.
Yeah, just one video. Just one video. Yeah. You just move.
And we're going to talk this morning about the immune system and cancer and how we're using artificial intelligence to make the immune system smarter and improve our ability to treat cancer. So I'm Stephen Grobmyer. I'm the chair of Fatima Bent Mubarak Center, which is Cleveland Clinic, Abu Dhabi. We're a freestanding cancer center which opened officially on March of 2023. We're modeled after the Tausig Cancer Center on the campus of Cleveland Clinic in Cleveland, Ohio, and many of us actually came from there. So I welcome you to this presentation. I'll start out by reminding you that cancer is a global problem, and we've made a lot of progress in treating cancer, but we have a long way to go. Results from cancer treatment have never been better. We have new treatments that have less side effects and are more effective than ever. But again, we have a long way to go. What I'm going to focus on today is how we use the immune system to treat cancer and how we're using artificial intelligence to make immune treatments of cancer even more effective. But to start, we're going to go back in time and we're going to look at how interest in using the immune system to treat cancer ever began. And we'll go through that. But before we go any further, I want to take you back. If you haven't been to medical school, we're going to go to medical school for just a few minutes, because it's critical that you understand some of the basics of cancer and you also understand some of the basics of the immune system so we can pull all this together. So I'm going to begin with cancer. And cancer, like I said, is a very common problem. Many of us have had cancer, or somebody in our family or somebody we know has had cancer. Well, it turns out cancer results from abnormal genes in cells. So something causes the genes in a cell to become abnormal. And there are various reasons why a gene in a cell would become abnormal. What genes do is they program the cell to make something called proteins. Abnormal genes in cancer cells make abnormal proteins. Some cancer cells have a lot of abnormal genes and they make a lot of abnormal proteins. Some cancer cells have only a few abnormal genes and only make a few abnormal proteins. So there's a very heterogeneous mix of genes and proteins in cancer. But that's what I need you to understand, is that abnormal genes lead to abnormal proteins. Some cells have a few abnormal genes and a few abnormal proteins. Some cells have a lot of abnormal genes and a lot of abnormal proteins, okay? The other thing I need you to understand is go back to medical school and understand a little bit about the immune system. So the job of the immune system is to protect all of us from things that don't belong. And in the main, that means bacteria, viruses, other infections, things like that. What happens with our immune system, it's constantly on. The immune system is going throughout our body and it's looking for things that don't belong. And when it identifies things that don't belong, it elegantly, in normal circumstances, mounts a response, an immune response, and that immune response attacks the invader, eliminates it from our body so we can go on and live a healthy life. What we're going to talk about today is how to leverage, or what efforts have been done to leverage the immune system, not only to treat bacteria and viruses, but also to treat cancer. And you're going to see that what's happening is we're trying to use the immune system to recognize those abnormal proteins, which I just told you about exist in cancer cells. Some cancer cells have a lot of abnormal genes and a lot of abnormal proteins. Some cancer cells have a few abnormal genes and a few abnormal proteins. So that's the basics. Basic understanding of cancer and abnormal proteins and genes, and a basic understanding of the immune system. The other thing I need to tell you about the immune system is it can be a double-edged sword. While it can elegantly eliminate things that don't belong in our body, if the immune system is too vigorous, it can lead to a lot of problems. And that's where controlling the immune system becomes very important. So the ultimate example of that is something called septic shock. So you may have heard of septic shock. It's when the body mounts a massive immune response to an overwhelming infection. And in the immune system's attempt to clear or eradicate the infection, there's a lot of side effects, which in some cases can be lethal. So we need a controlled immune response. If the immune response is too vigorous, yes, we'll destroy the invader, but in the process of that, the patient may not survive. And there are other examples of over-vigorous immune response, like ulcerative colitis, where the immune system attacks the intestine and causes lots of bad problems. Rheumatoid arthritis, same thing, causes joint problems where the immune system is out of regulation. So we need the immune system to be perfectly controlled to get the results that we want. So now we're going to go back in time. We have the background. We're going to go back in time. And the first question I want to ask or talk to you about is, how did the world ever get interested in using the immune system to treat cancer? So the story goes back to the 1890s in New York City, where there was a famous surgeon named Dr. Coley, and he worked in Manhattan. And he was a surgeon, a famous surgeon, but his specialty was treating a very rare and aggressive form of cancer called sarcoma. Sarcoma are still difficult to treat today. They were difficult in 1890 and they're difficult to treat today. A big part of sarcoma treatment still is surgery, removing the sarcoma. What happened to Dr. Coley in 1890 was he was operating on a lot of patients with sarcoma and other cancers, and he had a young woman who came to see him, and this young woman had a sarcoma of her hand. And the treatment of the day for the sarcoma was amputation, removing the hand. There was no evidence at the time he did the surgery that there was cancer anywhere else in the body. This is 1890. So he did what every surgeon did at that time, which was remove the hand to apparently cure the patient. Well, what happened was he did the operation, the patient recovered from the surgery. 4 months later, the patient returned, and it turns out that the patient had developed sarcoma in the lungs. And that this surgery, which was designed to cure the patient, didn't cure the patient, and she ultimately succumbed to cancer, which was on the hand, which had spread to the lungs. So very frustrated by this case and other cases, Dr. Coley did what a lot of us who are in the medical profession do. We start to ask questions. We want to do a study, a research study, to figure out how we might make things better for patients. So Dr. Coley went to New York Hospital, a large hospital in New York City, and he started to review all the records of patients who had had soft tissue sarcoma to try to see if there were any patients who had done exceptionally well, and see if he could identify any factors which were associated with a good outcome. And he searched through all the records in the hospital, and what he found was one patient named Fred Stein. And Fred Stein is a patient he had operated on who had a very large sarcoma of the neck. And he had gone through multiple operations to remove the sarcoma. And in most cases, in spite of operations, the patient would succumb ultimately to sarcoma from spread to the lungs or other parts of the body. But what he identified was Mr. Stein had survived. And in studying the records of Mr. Stein and all the other patients, what he identified was, he observed that Mr. Stein had had an infection, a skin infection, called Erysipelas, which is a skin infection caused by streptococcus, which is a common bacteria. So he was so intrigued by this, he didn't believe it. He actually went down from the Upper East Side of Manhattan to the Lower West Side, and he found Mr. Stein. This is 7 years after his last surgery. And this is a drawing of Mr. Stein, who you can see a scar on his neck where the surgery had been done, but he was alive and well. And he postulated that it was this infection which led his immune system to not only mount an attack on the strep infection, but also to mount an attack on the cancer. So what Dr. Coley did was he started treating patients by injecting them with bacteria or parts of bacteria. And he had some success. And this is something that he continued to do as a way to treat cancer using the immune system for about 30 years. After 30 years, other treatments were coming along, and this use of bacteria or parts of bacteria to treat cancer kind of faded away. But continued interest in using the immune system to treat cancer continued. The next thing that happened, fast forward several years, I'll take you to the 1970s, when the idea that a general stimulation of the immune system could be used to treat cancer was being developed. It turns out within all of us there are molecules which stimulate the immune system. One family of molecules which stimulate the immune system are called interleukins. All of us have interleukins in our body, and when our body needs to mount an immune response, interleukins get upregulated, and it tells our immune system that it has to activate and start to treat infection or do its job. So in the 1970s, the idea came along in Washington, D.C. and other places that for patients with advanced cancer, that a general stimulation of the immune system using IL-2, interleukin-2, this natural molecule in our body, which could now be produced, IL-2 would be given to patients with advanced cancer, and guess what? Some patients with advanced cancer were being cured by generally stimulating the immune system with IL-2. The problem, as I mentioned before, with general stimulation of the immune system, yes, certain patients were being cured of cancer, but also some patients were dying of an immune response which was out of control. So we had a new treatment for cancer which involved the immune system, but the problem was it was unregulated. While we cured some didn't survive. So we needed to do things better. And what happened after that was continued understanding of how the immune system works. And that's where we get to the next slide. So we're going to go back to medical school, and I'm going to talk to you about a very specific cell in the immune repertoire called the T cell. And if you're a student of cancer treatment or cellular therapy or immunotherapy, the T cell is a primary actor, okay? And let me explain to you how the T cell works, because it's important as we go forward and try to understand how AI is being used to treat cancer. Let me explain to you how the T cell works. So this is a T cell here. And on the surface of a T cell are these receptors. They're little flags. And T cells go throughout our body. And what the T cell's doing is looking for anything abnormal in the body. And when it sees something abnormal that it recognizes, which is here, recognizes, it mounts an immune response. You get an immune response. You generate cytokines. It stimulates a lot of other cells in the body to attack the foreign invader. If the T cell sees something it doesn't recognize, then nothing happens. So the T cell is going around. If it's not seeing anything that's abnormal, everything's fine. When it sees protein that it recognizes, then an immune response gets mounted. If the T cell sees a lot of abnormal proteins, okay, then a giant immune response gets mounted. And that's because there's many abnormal proteins floating around. The body, to clear many abnormal proteins, needs to mount a big immune response, okay? And so it was this understanding of the immune system that has become the basis for a lot of the treatments that we have. The other thing that's happened is beyond an understanding of the T cell, an understanding of something called the checkpoint system has also come along. The checkpoint system, it turns out, is a set of molecules which regulate the immune system. We now have drugs which can be used to turn on and off the checkpoint system. The checkpoint system controls the function of T cells, and the checkpoint system is the basis for the awarding of the Nobel Prize to Jim Ellison at MD Anderson several years ago. Because now we have drugs which specifically can turn on and turn off the immune system, these checkpoint inhibitors, okay? So we have an immune system, the backbone of which is the T cell. It responds to foreign proteins. Small amount of foreign protein, small response. Large amount of proteins, large response. And we have a checkpoint system for which we now have checkpoint inhibitors, which are drugs which are used every day in the clinic. And what checkpoint inhibitors do is they unleash the T cells to do their job. Okay? It turns out that checkpoint inhibition is a very effective treatment for some cancers. Okay? Very effective. So more effective than anything we'd ever seen before. And this is why Jim Ellison won the Nobel Prize, because he developed drugs that allowed us to turn on the immune system. So this is an example published in a famous journal almost 10 years ago. But this is a patient who had an advanced form of skin cancer. Normally, in the years past, untreatable skin cancer that almost always led to the death of the patient. This was published in a famous journal because this patient was given a checkpoint inhibitor, the drugs I mentioned to you, and it turns out that when the patient was given a checkpoint inhibitor, the cancer went away completely. This was a very powerful demonstration of the ability of using the immune system to treat cancer. Very important. In addition to checkpoint inhibition, we also have other ways we're leveraging the immune system to treat cancer. One way is through the use of vaccines. So it turns out that on certain cancers, there are certain proteins, abnormal proteins, that can be identified, and we can vaccinate patients, just like we do vaccination for COVID and flu and everything else. We can vaccinate patients against those proteins, and we can trick the immune system to attack cancers which express abnormal proteins. So that's happening. It's actually happening in the real world, and if you go to Cleveland, Ohio, where I was working before I moved to Abu Dhabi, there's actually a program in vaccinating certain high-risk patients for breast cancer. So we give them a vaccine with the goal of preventing them from getting breast cancer. So that's another example of using the immune system. And the final example of using the immune system to treat cancer is something called CAR T therapy. Okay? Let me explain to you what that is. We talked about the T cell before. So with CAR T therapy, what's done is the patient goes to a lab, the T cells are taken out of a patient, and they're manipulated or altered in the lab. And they are altered in a way to program the T cell to actually attack the cancer. Specific receptors are put on the patient's T cells. Those receptors are specific for the cancer. So the cells are taken out, they're manipulated, and they're put back in the patient. Another way we're using T cells for the immune system to treat cancer. Okay? But in spite of all the advances that we have in using the immune system and many other treatments of cancer, we still have a long way to go. And for many patients, immune therapy doesn't work. And I want to put all this together and explain to you why we think, in some cases, immune therapy doesn't work. So let me take you through this diagram. The far left column is a stop sign, okay? Red is here, you can't see the color, but these are red, these are green. If it's red, then the patient did not get a checkpoint inhibitor. And I just showed you the checkpoint inhibitors unleash the immune system and allow them to treat cancer, as I showed you in that picture. These are patients that didn't get the checkpoint inhibitor, okay? No checkpoint inhibitor. So a patient develops cancer, and I have two different types of cancer. I told you there's cancers that have a few mutations and a few abnormal proteins. So this is a patient, no checkpoint inhibitor. Actually, this is a patient with a lot of mutations and a lot of abnormal proteins. Lung cancer and melanoma are examples of that. But because there's no checkpoint inhibitor, the immune system mounts a response, but it's very weak and doesn't actually lead to the eradication of cancer. This is a patient with a cancer with a few abnormal mutations and a few abnormal proteins, but again, no checkpoint inhibitor, very weak immune response, no treatment of the cancer. Now, this is why we see an effect of the immune system on cancer here. So this is a patient who gets a checkpoint inhibitor, so we've now turned the immune system on. We have certain cancers like lung cancer and skin cancer, okay, which have high number of mutations, high number of abnormal proteins, and I showed you this is a perfect combination for the immune system to go and attack those cancers which contain those abnormal proteins. That's why checkpoint inhibitors work. That's why that case I showed you the checkpoint inhibitors had such a profound effect. We turn on the immune system. The melanoma in the picture that I showed you has a lot of abnormal proteins, so the immune system is then able to attack and eradicate the cancer. We have another situation where we use the checkpoint inhibitors, and in many cancers now, checkpoint inhibition alone is not enough. And it's because we turn on the immune system, but we have certain cancers like common breast cancer, where we don't use checkpoint inhibitors. It's because there's not that many abnormal proteins. So the immune system doesn't get stimulated. Even though we give it a checkpoint inhibitor, there's not enough abnormal proteins to stimulate an immune response and treat the cancer. This is why we are limited in what the immune system can currently be used to treat cancer. This is where AI comes in, okay? And I'm going to show you how we're using AI to make this whole process better, and how we are making the immune system more effective than ever using AI to treat cancer. And that's shown here. There's three primary ways that we do this. There's others on the horizon, but these are three main ways. The first is using something called radiomics. So in taking care of cancer patients, we do all kinds of imaging. And what's happening in radiomics is we're not just having a radiologist look at an x-ray, we're having a computer or an artificial intelligence system look at an x-ray. A single digital image will have millions and millions and millions of data points, which is far beyond the comprehension of any single radiologist. But it turns out if you use artificial intelligence to analyze the medical image, the digital medical image, you can create signal. And what happens is we can go and look at patients and compare their imaging to historically whether they responded to immune therapy. And then we can use artificial intelligence to develop imaging predictors of who will respond to immune therapy and who won't. Okay? And this is important because some of these immune therapies have toxicity. And if I have a lung cancer patient and we do an image of the lung, and artificial intelligence analyzes the image, and they tell the doctor, based on radiomics and the interpretation of the digital image, this is a patient who won't respond, there's no reason to give the patient treatment. On the other hand, if we have a patient who maybe traditionally type of cancer doesn't respond to immunotherapy, we can use radiomics and we can say, hey, based on the digital signal from this imaging or these images, we can predict this is a patient who will respond. We can give the patient immunotherapy and expect a good response. So that's just one way. The second way, the last two ways go back to some of the biology that we talked about. We talked about vaccinations briefly before. So what's happening now in the world is we do surgery or biopsies on patients with cancer. We then analyze the tumor once it's been removed or biopsied, and we sequence the genes within the tumor. We then also sequence the patient's immune system, and we can use this artificial intelligence to predict which of the proteins on a patient's tumor will elicit the biggest immune response, and we can develop a vaccine for that patient's tumor to give that patient. Because there may be many proteins that are abnormal on a cancer cell, but it turns out that certain ones of those abnormal proteins turn out to be better candidates for vaccination than others. So we do this type of analysis to determine what protein in a patient's tumor to create a vaccine with. And this is work that's ongoing in certain parts of the world, and is a very bright spot in the future of cancer treatment. And then finally, we talked about CAR T cells. CAR T cells, remember, patient has cancer. We take the T cells out of the body. The T cells go to a lab. We manipulate the cells and we put them back in the patient, okay? So what's happening now with artificial intelligence is you can predict how to engineer this receptor you're going to put on the CAR T cell so that it's most able to attack the cancer, okay? So this is analyzing the abnormal proteins on a cancer, figuring out what structure of a T cell receptor would be most efficacious in allowing the T cell to attack the cancer once it's put back in the body, and we engineer T cells and deliver them for cancer therapy. So it's a new way to use CAR T or T cells removed from the body to empower them to go and attack the cancer. So hopefully today you've learned something about the immune system, how we got started in using the immune system to treat cancer, and what the future looks like in terms of building upon the massive progress that's already occurred in the world to use artificial intelligence like we're doing at Cleveland Clinic Abu Dhabi with our partners at G42 to better understand the immune system and the response to abnormal proteins and abnormal cells so we can make the treatment of cancer better with less side effects to improve the lives of our patients. So thank you for your attention. I'm happy to take any questions that you might have. Question? I can't hear.
It's already on. Yeah, you can hear from the VA there.
Okay.
Thank you very much, Dr. I'm very happy to see a medical doctor talking about AI. I'm doing the same. I'm a computer scientist in my background. I'm trying also to learn. I'm fascinated by this field. I'm part of a G42 company called Inception, where we are also very actively looking at foundation models built on DNA, on protein, on chemicals for drug design as well. So thank you very much for the pedagogic effort you made to make everything simple, to understand cancer, to understand the immune system, and so on and so forth. But still I have a few questions. So we all know that to train AI model, we need the tool, which is the data. So from where you got your data to train, there's three different AI models on radiomics, on proteins, and also on T-cells engineering. Second, how in healthcare in general and this kind of projects, how you are controlling the use of AI responsibly and safely?
Good questions. Thank you. The first question was about
The data.
the data. Oh, so the data. The first question was about where do we get the data to do this work. So we're fortunate, and I've been very fortunate over the last 4.5 years since I moved to Abu Dhabi in 2019 to be part of something called the Emirati Genome Project. And it turns out the Emirati Genome Project is one of the largest national genome strategies in the planet Earth. And to date, over 600,000 Emiratis have volunteered to have blood drawn and have whole genome sequencing. So when you want to begin to understand what the T cell receptors are in a population of patients, you can turn to resources like the Emirati Genome Project. We're also working with our partners at M42 and the Department of Health on helping, hopefully in the future, to develop a national biobank. National biobank, or biobank in general, is a concept where patients who have surgery or biopsies, part of their tumor is stored within a central repository, and as part of that, DNA sequencing of the tumors are done, and that becomes very useful information for doing work like this. So we're actually very uniquely positioned here with some of the resources that we're blessed to have around us to do some of this work, and it's work that's ongoing. We're also working with our partners at M42 to use AI in other ways to improve healthcare. For instance, working on unraveling the national medical record called Malafi, because there's tons of information within the electronic medical record that we believe with artificial intelligence we can extract predictors, for instance, of people who will develop certain diseases, or better understand how patterns of disease are occurring, or what treatments in aggregate populations of patients are effective. In terms of how we protect the use of AI, this is a great question. We work under the auspices in my hospital and most hospitals under the auspices of an IRB. So that's one way it's done in terms of our own institutions. We also, for work like this, are in general under the regulation of the regulator, Department of Health. For many of the projects we do that involve AI or genomics, is very involved in the approval process of making sure that we're using the information in a responsible way. We call that in healthcare de-identified, or protecting the identity of patients. It's done without question, and it's all goes through an approval process before we do that. But it's a very important part of responsible use of this information, which can be obviously very powerful, but you're right, if used in the wrong way, can also be bad for patients. So thank you for the questions. Yes.
Thank you for a very interesting talk. Similar to my, I come from computer background also, maybe medical, and I really like this talk. I'm curious on some description you provided on, so if we use immune system to treat cancer, what stops the abnormal proteins to turn again and recur cancer?
Yeah, that's a great question. So what happens with these proteins is, and the reason the immune system recognizes them, is the proteins that the genes produce in general are, a lot of them are put on the surface of the cancer cell. They become part of the cell, because in biology, what determines a lot about the function of a cell and how it interacts with other cells are the proteins on the surface of the cell. There's receptors, there's all kinds of modulators and things on the surface of cells. So what the immune system is recognizing is actually abnormal proteins on the surface of cancer cells. And the job of the immune system in these cases where it's attacking the cancer cell is to eradicate and destroy all the cancer cells. Like that skin cancer case I showed you. You turn on the immune system, a massive specific attack against the cancer occurs, and the cells, the cancer cells, which contain the abnormal DNA, which are making the abnormal proteins, are eradicated by the immune system. So there is no more cancer. And there are clearly cases that using immune therapy, we see patients who historically would never be cured of cancer cured. And that by definition means that all of the abnormal cells have been eradicated from the body. Once you eradicate the abnormal cells from the body, no more cancer. Now you're right. What caused the cancer in the first place? In many cases we don't know, but many patients who are cured of cancer never develop cancer again. So there are reasons that we may not understand, but good question. Thank you. Thank you very much for your attention, and I'll be available after if there's any more discussion or questions. Thank you.